All right, so thank you and welcome to this I3 webinar. It's all about the future of freezing. It is sponsored by Future Fertility. Future Fertility has developed the first non-invasive AI oocyte assessment tool. They're leading the industry with the largest and most robust data set to provide objective, standardized, and repeatable assessments of oocyte quality. Future Fertility's AI-powered tools are research-backed, equipping fertility experts and their patients with validated, personalized reproductive insights. This webinar today focuses on cutting-edge cryopreservation techniques. We'll be discovering new groundbreaking strategies in oocyte and embryo vitrification and warming, and we'll dive into the intricacies of cryopreservation theory and practice, exploring innovative methodologies. Additionally, we'll explore the current landscape of ovarian tissue freezing and vitrification, and we'll receive updates on the intriguing method of lyophilization of germ cells. Can't wait. I'm joined here by my co-host and one of the co-founders of Future Fertility, our Chief Medical Officer, Dr. Dan Nayot. Dr. Nayot is a practicing reproductive endocrinologist and infertility specialist at the Reproductive Care Center in Canada. He is also the medical director at the Fertility Partners and North American network of IVF clinics. Dr. Dan Nayot completed his BSc in mathematics at the University of Toronto, an MD at Western University, and Masters of Clinical Epidemiology at Harvard, his FRCSC in OBGYN at the University of Toronto, and is an REI Fellowship at McGill University. He is committed to innovation and data-driven research to optimize the patient experience and expand access to fertility care. With Future Fertility, Dan brings his extensive knowledge in medicine and mathematics together to solve gaps that he has experienced in over a decade of clinical experience. So thank you, Dan, for co-hosting with me.
Thank you. It's a real honor to co-host. For those who have not met Jullin yet, it's a real honor. Jullin Fjeldstad began her fertility career at Victoria Fertility Center in Canada in 2007, and she quickly became a senior embryologist and successfully held the position of a lab director for several years. She has a BSc in biology from the University of Victoria, Canada, and a Master's of Science in Clinical Embryology from Leeds, UK, achieving distinction in both areas. She's always had a passion for innovative arts aimed to improve the fertility journey and patient IVF outcomes. Today, I'm honored that Jullin is she is the head of the clinical embryology and scientific operations for Future Fertility. She works closely with partnering clinics to ensure smooth onboarding of innovative AI-based tools within their laboratories. Additionally, she oversees all our scientific project collaborations, and you may have seen her amazing manuscript just published recently at RBM Online. For everybody here, you can find all the amazing I3 webinars online for free. Just go to ivfmeeting.com. Certificates are great. You're going to get your certificates, but you got to give us a little bit of time. So they're not going to be immediate, but we do promise you'll get your certificate within a few days. We want this to be interactive. There's a Q&A. So there's chat and there's Q&A. Please go to the Q&A. That's where you'll type in your questions and we'll field it at the end. So please stay with us till the very end. So that's when we're going to get pretty interactive. So we've got 3 speakers and let's get started. It is an honor to introduce the first speaker tonight. Dr. Juergen Liebermann has been working in the field of IVF since 1995. He is a director of laboratory services in Chicago-based fertility centers of Illinois that's partnered with the U.S. Fertility, Ovation, and RMA New York network. He received his doctoral degree from the Technical University Munich in Germany in '95 and postdoctoral thesis in 2004, which qualified him as a university lecturer in experimental reproductive medicine at the Bavarian University of Würzburg. He contributed more than 160 oral presentations at both national and international meetings and has authored more than 66 abstracts, papers, and book chapters in reproductive science. In 2007 and 2016, he co-authored a book with Dr. Michael Tucker titled Vitrification of Human-Assisted Reproduction, and he is a member of ASRM, ESHRE, and ABB, and that's why he is our speaker tonight. He will be presenting fast and furious new strategies in oocyte and embryo vitrification and warming. Thank you.
Thank you very much. I would like to thank you, the International Initiative, to invite me to this webinar. And I would like to use the next 30 minutes to update everyone who joined us today on new strategies in oocyte and embryo vitrification, as well as warming. Okay. My first slide, it will show you a little bit the outline of my 30 minutes. First, I will talk about the blastocyst and how you can implement it quite quickly in a clinical application setting. And my second talk is what I still consider research on human oocytes, vitrification and warming. When we look at the recent data from SARD about published 2021, and we compare 2020 to 21, we see increase of IVF cycles of about 67,000 cycles. If you would look at the number of 4 cycles, we see about 14,000. And if we look at 4 cycles for donor and embryos, about 4,000. And for cryopreserved oocytes, is it embryo or oocytes, about 15,000. So altogether, we have an increase of about 20,000 cycles in 1 year. So there is an increasing demand in the next couple of years in terms of cryopreservation. It is also estimated that we get about a 10-fold increase in IVF cycles in the next couple of years. And so we have to sit back, and have to look at our protocols, if they are still efficient, and is a way maybe to improve them by even improving our success we already achieved. When we look at the evolution of vitrification, it starts actually with a landmark paper by Rollin Fay in 1985, when it was the first publication of a live birth from a vitrified oocyte by Kuleshova in 1990. And then after 2000, we got endless papers on day 3 and blastocyst vitrification. And so over the years, we got a lot of publications on this topic of vitrification, that also ends up in 2 books we presented in 2007 and 2015. But if we look at the protocols, we didn't really change much. We increased the volume of the drops, and we moved away from merging the drops, and that's pretty much. Our solutions, in contrast to this, are still the same. You know, we still have a Hebes buffer base media with 20% serum, and then we work with an equilibration solution that's usually 2 cryoprotectant. It could be ethylene glycol or DMSO replaced by propandiol, each 7.5%. So our ES would be 15%. And then a final step in the vitrification solution for only a short moment, that is again 2 cryoprotectant now with a concentration each of 15%, that would bring the solution to 30%. In the final solution is also a 0.5 molar sucrose. So in May, I was sitting down in 22 and thought about, also a couple of questions, what we can improve on blastocyst vitrification and warming. Is the time of the procedure the right one? Do we use the right solutions and concentrations? Do we need to dilute sucrose at warming? That's actually a central dogma of the cryobiology. If so, does it always have to be 1 molar sucrose? And finally, for blastocysts, at least after warming, do we need to culture in media supplemented with 20% protein until transfer? What we know so far is that a stepwise removal of the cryoprotectant and rehydration in serial osmotic solutions to prevent osmotic shock has been a fundamental principle in the cryobiology for decades. The theory behind this approach is to gradually eliminate the cryoprotectant and rehydrate the embryo to minimize damage during the vitrification warming processes. The entire process is time-consuming and labor-intensive in each IVF laboratory. It is evident that a protocol with 90 to 100% survival using a single-step solution without stepwise procedure would be challenged for the central dogma of cryobiology. And we always look out for new publications. And so I relied here on 2 where I was building up my research. It was a group in 2021 and 2022, they achieved a prize paper as ASRM and PCRS. And finally, a paper about oocyte vitrification by Gallardo in 2019. So these were my two basic references. I started to do research. And so we started with blastocysts first, and we did an intensive in-house trial on embryos, and finally on oocytes. And that's our in-house trial on blastocysts before we moved it in a clinical setting. We used embryos where had a disposition for discarding signed by the patients for research. And we fought 564 blastocysts. We split them off in biopsied blastocysts and non-biopsied blastocysts. And what you see in the second column is the survival right after warming. We kept them in culture in 20% media until 24 hours later, and we looked again how they present themselves, and you see still a good survival. And on the end, we finally saw about 67% of these blastocysts hatching or hatched. Remarkable was that we observed a much lower rate in non-biopsied embryos. About 60% of the non-biopsied embryos hatched on their own, whereas in the biopsied embryo group, it was close to 88%. Based on these results, at least for us, we still feel considered to assisting hatching, at least on non-biopsied embryos. And this is now our protocol we implemented in September of 22. It is one single drop of 400 microliter TS, of course, at 37 degrees. We plunge our carrier in this solution, take it from there and move it in the transfer dish, that is usually an organ culture well. We rinse the blastocyst briefly outside, and then we keep it in the center well until transfer. And this is so far our results. Positive beta first. We did 2,296 transfers, and you see based on age, besides only patients over 40, positive beta is consistently over 70%, 73 in average. When we look at ongoing pregnancy, and this is based on 2,100 transfers and implantation, you see here you can expect ongoing pregnancy of close to 60% for under 35, with a 64% implantation. Will be dropped with the age, but still, if you go to 38 to 40, it's still 55% ongoing pregnancy with 62% implantation. We looked also, besides the age, at the day of development, day 5, day 6, day 7, and you can, if you have day 5 for transfer, expect an ongoing pregnancy of about 59%, that drops with day 6 embryos to 51, and very similar for day 7, around 50%. Implantation is 64% for day 5 and 57.6 for day 6. Of course, we looked at untested and euploid embryos, and you see here a benefit in doing PGD. It's a 51% ongoing pregnancy and 58 for euploid embryos. And finally, we looked at the age and the decision in doing genetic testing and not doing genetic testing. And if you look at each age group, for example, under 35, we still see here a 70% increase in implantation for under 35. That even gets more significant if you do testing, if we get to patients with advanced age. For example, over 40, it's 46% if you have an untested embryo, or it's 56.5 implantation rate for over 40 if you test the embryo. A picture on the right is a blastocyst we rehydrated for 1 minute, and we usually do transfers 2, 3, or even 4 hours. In that case, was it 4 hours? We kept in culture until transfer. Our live births, we can look at 723 transfers so far, and I compared that to about 3,000 cycles we did between 2007 and 14, and we see a significant increase in the live birth. We achieved 31% in the early days, and now with this protocol, we can expect a live birth of 51.5%. Average weight of the children is similar and for totally normal also as well. It's a average pregnancy duration. We also increased with the protocol over years, of course, our rate of singleton. We now close to 98% singleton and only about 2% twins. When we also had the question, with 4 in 1 molar, is maybe a chance to even lower the sucrose. Now is also one publication from Belgium under the group of Dr. Gonst. They showed that actually you can go lower to 0.5 or even 0.25 molar. And we confirmed that also with an initial trial here in-house and presented that at the last ABB meeting in Las Vegas. There is a trend forward early resumption of functionality is observed after warming in low sucrose medium. For example, Gans observed that the time blastocysts need to hatching if you thaw them in lower sucrose only 5.1 hours compared to about 7.8 hours if you thaw them in 1 molar. And this is some blastocysts we thawed, non-biopsied, and we cultured them in 10% instead of 20%. And the next slide is embryos, biopsy blastocysts. We also thawed all in 1 minute, and they were cultured also in 10% and not in 20%. So to summarize to that point on blastocysts, this protocol makes a laboratory technique easier for warming blastocysts. It simplifies. It reduces unnecessary time blastocysts are exposed to room temperature, and in turn reduces the stress to the blastocysts. There is less time for radical oxygen species creation and their impact on the embryo development. A lower sucrose, 0.5 or even 0.25 molar, is possible, as well as culture in lower protein concentration. Short dehydration as well as short rehydration is easy to implement in your daily routine and standardize your blastocyst cryopreservation procedure, and its outcome even more successful. The one-step warming protocol is superior to the multi-step protocol in terms of pregnancy outcome, efficacy by supporting an optimized workflow, as well as cost effectiveness. So what we just learned on Fast and Furious of blastocysts. We have demonstrated that a gradual removal of cryoprotectant and a stepwise rehydration are not necessary for the survival of blastocysts and the ability to regain their development. The one-step short rehydration protocol will reduce cost. We estimate about 40% and labor of about 95% in any IVF laboratory. Now I would like to come to my second part, and as I mentioned before, this is still research. It is how we can improve oocyte freezing in terms of survival outcome, but also shortening the time. We know that oocyte freezing is an important aspect in every IVF center. There's a couple of reasons why we freeze oocytes. For example, no sperm available on day of egg retrieval, poor responder where we use multiple cycles to support oocyte accumulation, egg donation, of course, it's more convenient, gives the patients more independency, it's more flexible and time saving. We have also patients, of course, with fertility preservation in cancer patients. And then it's age-related fertility loss, social freezing for advanced age, and in cases of premature ovarian failure. And this is our in-house numbers that also shows you clearly that our freezing cycles in regards to oocytes goes up consistently every year. And also my post I posted last year in November, and the interest in this demonstrates how many people still are interested to improve these protocols. So in 2024, egg freezing is definitely safe. Data are reliable, repeatable, and successful. However, is there a missing piece of the puzzle regarding oocyte vitrification as well as warming? And so I went, as I did similarly for blastocysts back, and had a couple of questions. What you usually do when you want to start with a research. Is the time of the procedure, is it correct? Yeah. Do we use the right solutions? Do we need to warm in 1 molar? Do we need to stick to 2 oocytes to load per carrier only? If we can load more than 2 oocytes, will be increasing volume compromise the improvement? Can we freeze all developmental stages with the same protocol? And that would include also cleavage stage embryos and zygotes. We all have media from different vendors. Do they deliver comparable results? And if so, are these media interchangeable? And finally, again, do we need to culture in 20% protein post-swarming, or does 10% work as well? And this is just a reminder how most of us freezing oocytes. This is the first 3 minutes in ES. The oocytes is shrinking because it's releasing water. Cryoprotectant is penetrating the oocyte. After 6 minutes, you see already a regain of the volume. And when we come to 10 minutes, the oocyte achieved a regain of about 80–90% of its original volume until we expose them to the final stop of VS for about 60–90 seconds, and we see a dramatic shrinkage again of these cells. However, based on a paper by Gallardo and his colleagues in 2019, they found that oocyte osmotic behavior indicates that the dehydration upon exposure to standard cryoprotectant solutions occurs very fast. The point of minimum volume of the shrink bell curve is reached within 60 seconds. At this point, intracellular water ejection is completed, which coupled with the permeation of low molecular weight cryoprotectant results in similar intra and extracellular solute concentration, what we will call isotonic. That means that prolonging the exposure to the cryoprotectant does not improve the cytosolic class formation tendency and could be avoided. After 2 minutes of exposure to standard cryoprotectant, the critical intracellular solute concentration necessary for successful vitrification was attained. And so I started my research in May of 22, and it took me about 5.5 months until I found the protocol I want to present here, what I consider as successful. And this is my protocol, how I freeze oocytes, GVs, metaphase 1, metaphase 2, zygotes, as well as cleavage stage embryos. We start with a drop of VS, briefly rinsing the oocytes. When I put them on top of a 200 microliter drop of ES, let them sink. It takes about 30 seconds for the cells to reach the bottom of the dish. And after 1 minute, I went through 2 solutions of VS, each 100 microliter in 1 minute, when I load them and then plunge them. So the whole protocol takes 2 minutes. The warming, and this is a very critical step. I don't start with 1 molar sucrose. I start with DS, 0.5 molar sucrose. And I usually do 500 microliter drop. I put the eggs in this drop for 1 minute. After 1 minute, I carry a little bit over DS in the first drop of WS, wash solution only. And after 30 seconds, I move them finally in wash solution. There should be no sucrose anymore. And when they go in a culture dish with global high protein, 20%. So warming also takes me 2 minutes. So I started comparing 2 protocols first. I started with Irvine and Vitrolife, and you see here my GV numbers and their survival as well, metaphase 1 and their survival. And you see, at least between these 2 media, there is no significant difference in terms of survival if we consider GVs or even the survival of metaphase 1 oocytes. Of course, I didn't stop at this moment just to consider them as survived. I cultured them in 24 and even 48 hours just to see if they also can regain their meiotic activity. So out of these 565 GVs, 95% survived. 24 hours later, I had about 92% converted to metaphase 1, so a breakdown of the German vesicle. And then finally, about 55% of them converted to metaphase 2. I did the same with the metaphase 1. I kept them for 24 hours only in culture. And after a nice survival of 95.5%, about 85% of them converted to metaphase 2. As a control group, my control group is not as large as my study group, but 56 GVs I kept in culture, and I looked at them and their conversion rate, and you see this 94%, close to 95% converting to metaphase 1 compared to 92, and their conversion to metaphase 2 is about 60% to 55%. That's the same for metaphase 1, 19, and they converted close to 90% versus 85% for Savon vitrified. Here's just some examples. Metaphase 1 oocytes, remember they were vitrified in 2 minutes and 42 minutes. And on the right side, I have vitrified GVs. You see the German vesicle. And when I just magnify the picture, you see how clear and healthy these German vesicles are presenting as they were not cryopreserved. I did also use material that also is not for use for patients on fertilized metaphase 2 oocytes, or even metaphase 1. They converted overnight, but we didn't do ICSI because we were not ready for ICSI. And you see also here, again, two media, I compared more than 90% survival. I used 1 and 3% abnormal fertilized zygotes. They survived 200% as well. Cleavage stage embryos derived from abnormal fertilized oocytes. When I used Kitasado on sage, I did the same approach, different developmental stages, and you see the numbers were comparable, even similar to what I showed you before by using Irvine or vitrolife media. Here again, unfertilized metaphase 2, vitrified, and 20 hours later. And you see them here also. You see his nice polar body, very well preserved after these freezing and warming steps. This is also now vitrolife media cultured in high protein, 11 oocytes, all survived. And I started also, as I mentioned before, many of us receive embryos or eggs from somewhere else, and we usually don't have this medium in-house. So I was asking myself, what is this, are these media interchangeable? And, you see all these combinations here I actually undertook. I vitrified, for example, in sage, and when I warmed these eggs, I split it in sage or vitrolife. Or I vitrified in Irvine and thawed in sage, or vitrolife and Irvine. So I did all different combinations, and I want to just give you a couple of examples. For example, here I vitrified in Irvine and warmed in vitrolife, and you see GV is 100%, metaphase 1, 100%. You see the pictures on the right after warming. Another combination was freezing, vitrifying in Irvine, and then thawing in sage. And you see here I did 16 M2, 1 died, so 15 survived. And finally, also I tried sage as vitrification solution, and when I thawed in vitrolife, here I have GVs. When I have metaphase 1 died out of these 3, and when I had metaphase 2 oocytes, all of them survived in the next picture. Here we go. Then I had here Irvine solution. I cultured them now not in 20, in 10%. This is all GVs. I kept them, for example, in culture, as I mentioned before. And you see the conversion of these GVs to metaphase 1. And finally, to a high percentage, in this case, it was 65%, they converted to metaphase 2 using the same protocol, 2 minutes vitrification, 2 minutes warming. And I also want to show you how efficient this protocol can be if you vitrify a large amount of eggs. In this case, I've vitrified 40 eggs. I put 5 on a cryolog and 6 on 2. And when I started vitrifying, and it took me 12 minutes and 20 seconds to vitrify all these 40 oocytes. And we see when the survival here, 4 didn't survive, so 36 out of 40. And I usually start with 12 eggs, 2 carriers with 6 each. And you see it took me 10 minutes roughly to thaw these 40 oocytes. When we talk about more than 2 oocytes, so 4 to 6, people, of course, asking what about the drop size? And you see on the left side is a cryocarrier with precisely exactly 1 microliter pipetted on the cryolog. And on the right, you see a drop loaded with 6 GVs that is less than 0.5 microliter. And what about fertilization, embryo development, and outcome? And I rely here on a LinkedIn post by the lab director, Birol Adin from Ovogene, and he used that protocol pretty much in freezing 2 and 1 minute thawing. And you see here, his survival rate 100%, fertilization over 90%, cleavage rate was close to 90%, blastocyst formation rate about 57%, and his rate of euploid embryos after genetic testing was more than 61, close to 62%. He presented a clinical pregnancy of 65%, and all these eggs are donor eggs because these numbers from Ovogene are donor egg bank. So to come to my summary here and ask my questions, time of procedure, he can cut down the total time of 27 minutes to 4. When I say 27 minutes, we vitrified in 16, and when we thawed in 11, now it's 2 and 2. Do we use the right solution and concentrations? Yes, but for warming, I use 0.5 molar instead of TS. Do we need to stick to 2 oocytes loading per carrier only? No, we can load up to 4 and plus X. I would recommend 4. If you can load more than 2 oocytes, will be the increasing loading volume, compromise the improvement. As I showed, the volume, even with loaded 6X, is still smaller than 0.5 microliter. Can we freeze all developmental stages with the same protocol? Yes, we can vitrify and warm all these stages with the same protocol. We all have media from different vendors. Do they deliver comparable results? Yes, as I showed you. Is the media interchangeable? Yes, with those protocols showed here, media are interchangeable. And finally, culture media post-warming. Yes, meiotic activity is resuming in culture media with 10% or 20%. Thank you very much for your attention, and I'm looking forward to your questions.
Thank you, Dr. Liebermann. That was an impressive presentation. I love how methodical all of your research has been. I think as you've gone through this, you've answered a lot of the questions that have come up, but there's still numerous questions to be answered. I think everyone that's in the embryology space wants to learn your techniques and how we can, save the time, because we're all experiencing, these influxes of vitrification, for eggs and embryos. We have a question, I think time for one question at least, and then we'll jump into some of the other talks today, but we'll have lots of question and answers at the end. One question that's come up here, we have Salal from Turkey. I apologize if I'm saying your name incorrectly, but they say, thank you, Juergen, for the fantastic presentation. And they are asking, are you experienced with any zona hardening during this procedure? So is this similar to standard vitrification? And do you always use blastocyst collapsing for the embryos?
2 very good questions. So first one, this protocol does not change or prevent zona hardening because exposure will be to a low temperature of the liquid nitrogen. So we have to deal with the zona hardening as we did before with different protocols. In terms of blastocysts, this is a good point. Yes, we do collapsing, assisted collapsing. For every blastocyst we vitrify. If it's not a blastocyst we preparing for biopsy, they will then anyway collapse because of the biopsy procedure.
Perfect, thank you. So thank you for that presentation. I think we'll get on to our next speaker just to stay on time, and then we'll get back to more questions after. I'm excited to introduce our next speaker, Dr. Pierre Comizzoli. He started his career more than 30 years ago as a research veterinarian in South America and Africa. He then completed a PhD in 2000 on in vitro fertilization of bovine and deer species. In 2002, he joined the Smithsonian National Zoo and Conservation Biology Institute in Washington, D.C. to develop a new cryobanking project on gametes and gonadal tissues from rare and endangered species. He also coordinates reproductive monitoring and artificial inseminations in different species, including giant pandas, clouded leopards, or elk deers. Since 2007, he is the program leader of a Smithsonian-wide initiative to improve the management of and use of frozen biomaterial collections within the institution. His whole bio will be available on the website for further information, but I would like to introduce his talk today is going to be on long-term preservation of germ cells and gonadal tissues at ambient temperatures. Go ahead, Dr. Pierre.
Thank you, Jullin. Hear me? Yes, I assume yes, you can hear me. Thank you so much for the invitation. So I'm a little bit an outlier here because I'm not going to talk about cryopreservation. I'm going to talk about something that we may use in the future, which is storing for the long term at ambient temperature. Of course, everything that I'm going to be talking about right now is purely experimental. We are not at the stage where we can translate that into fertility clinics or into veterinary practice. But you will see that there is a lot of promising and encouraging results. So first of all, I do not have any conflicts of interest to disclose. Well, since we are talking about the future, I would like to remind you that our friend and colleague Bill Roll passed away last January, and he was definitely a pioneer who helped us to envision the future because, as I mentioned at the bottom of the slide, back in 1985, he published his very breakthrough research about vitrification. And that was really the first time that mouse embryos were preserved that way. And that really opened the door to a lot of application. And some of the things that Dr. Liebermann just talked about are really inspired from Bill Roll research. So thank you, Bill Roll, for your contribution, and we all miss you. So right now, when we talk about biobanking or cryobanking or genome resource banking, we're talking about really organized collection, storage, access, and use of those biomaterials for, of course, in your case, fertility preservation, but also in terms of to use in biomedical science. At the zoo in mice discipline, we use that for species conservation, but also, of course, for sustainability of food production. Right now, we store, of course, a lot of germplasm, reproductive tissues, cell lines, blood product, DNA samples. Everything that we store, I mean, we have just in North America, we have millions of samples that are stored right now at freezing temperature. And as you can imagine, in terms of the maintenance, the curation of the samples, it costs a lot of money. So right now, also, we need to think of what are the risk and the limitation also of the liquid nitrogen storage, especially for germplasm, because as you know, they require constant monitoring, they require specialized facility. Even for ultra-low freezers, it requires a lot of power. And all those requirements are not necessarily possible in many regions of the world. And unfortunately, as you know, also some of those containers, they may fail either because of human error or just equipment breakdown. So we really need to start thinking, I mean, not necessarily dismissing what's been done already with all those biobanks, but really to try to think of maybe alternate solutions that are simple, flexible, and cost-effective. So for that, and this is something that I'm not the first one to think of, and a lot of colleagues have been looking at around, the lessons that we can learn from nature. And in nature, there are organisms like tardigrades on the picture, or some plants or some fish embryos that are really able to sustain and suspend their life at non-freezing temperature. So this is providing them, this kind of a natural protection for dry, hot, cold, or really high salinity environment. But in terms of drying, I mean, in terms of, instead of talking about cryoprotectant, we can talk about xeroprotectant, really what makes, those organisms resistant to dried conditions. And of course, the trehalose is a natural sugar that's been very well studied and that is present in a lot of those organisms to resist dry conditions. And then after that, there is lots of different proteins that have been also studied that are also present in seeds, for example, that are able to stabilize the structures and all the biophysical, systems in the cells during this live suspension. So when we're talking about dehydration and desiccation, well, this is a little bit like, you can think of the same approach as the cryopreservation. So first of all, you need to remove the water. And then as you remove the water, you need to replace also the water with, in that case, so that's what we've been doing in our lab, is replacing the water with trehalose. What you are hoping to reach is that when you have a very low level of water, then after that, the trehalose has the capacity to vitrify basically at ambient temperature and forming this kind of a glass, exactly what you would have in a vitrification at ultra low temperature. So what we do is that we incorporate the trehalose. Unfortunately, in mammalian cells, well, we have to permeabilize the membranes, which is a little bit constraining, because the trehalose is not a sugar that can cross the membranes. Then after that, we have to remove the water, as I said, to reach really this vitrification or desiccation at ambient temperature. And then, of course, you can store, in theory, your samples at non-freezing temperature. But of course, you have to make sure that there is no contamination with humidity, because the slightest increase in humidity in your samples, then after that is going to destabilize everything, and you're going to have a degeneration, and you're going to lose the quality of your sample. And then after that, when you have to use your sample, just a simple rehydration is enough to reanimate everything. So right now, there has been a lot of techniques to remove the water after exposure to trehalose by lyophilization, which is, as you know, freeze drying. So you freeze the sample, and then after that, you expose the samples to a vacuum. So under low pressure, the water turns from ice to gas, and you have a sublimation of the ice, and you remove the water that way. You have also a lot of studies that have been done by passive air drying, evaporative drying, convective drying. But what we have chosen to explore in our laboratory is microwave assisted drying. Then here is just an example of the different storage device that you can have, but I will show you here more examples of what we are doing in our laboratory. You may be familiar with really the first successes with sperm cells, and Dr. Yanagi is really famous for a lot of things, but including also being able to, in his lab, to freeze dry for the first time mouse sperm, to inject them into oocytes, fresh oocytes, and then to have embryos, to transfer the embryos and to obtain live offspring. So that was a huge breakthrough, because it was a very good proof of concept for the fact that we can preserve sperm cells in these glass ampoules just for the long term. Even more recently, that same team has been able also to ship by mail just dried sperm from lab to lab, and to show that the sperm is still viable after it is injected into an egg. So instead of, in terms of other species, my colleague Pasqualino Loi in Italy has also, and in collaboration actually with Amir Arav, who will be speaking just after me, they've been also studying, the same kind of approach, but for other mammalian species like sheep, and they have also obtained very interesting result in lyophilizing sheep sperm. So what we are doing here in our laboratory, it's a little bit different. It's not lyophilization. As I said, it's microwave assisted drying. So what we do is that we deposit the samples after exposure to trehalose. We deposit the samples into those small filter papers or into those small glass cover slip. They are then after that placed on the rotating on a turntable that goes into a microwave. This is a microwave that has been customized. Of course, you don't want to cook the samples. You just want to expose them to pulses of microwaves that are going to be exciting the water molecules, and then with the heat are going to leave the sample. But of course, you don't want your sample to go over a physiological temperature. So there is a program on this microwave that helps to just have the samples that stay within a physiological temperature while they are getting dehydrated. And then once the beauty, the advantage compared to air drying is that the evaporation is much more homogeneous in your sample than with simple air drying. But then importantly enough, as I said, during the storage, you need to use those sealed pockets that are made of mylar, and where, of course, you prevent any contaminations from humidity. So what we started to do using the domestic cat model is really where we were really interested in trying to find some alternative to oocyte cryopreservation, because we have a hard time to cryopreserve oocytes, not only in the domestic cat, but in a lot of wild cat species. So we started to use the domestic cat as a model, and the germinal vesicle oocytes also as the experimental model. But instead of trying to dry immediately the whole oocyte, that, as you know, is a very complex cell, it's very large, it contains a lot of water, we concentrated our efforts on the germinal vesicle. And here, this is really to show you how it works, because when you dry, as you can see, when you look at the black dots here, the black curve, when you expose the samples to different times of microwave pulses, at some point you reach a very low moisture level. But as I said, the advantage is that in the presence of trihalose, you increase also the possibility to store your sample at, here, for example, at supra-zero temperature and even at ambient temperature. But you need really to go very, very, very low in terms of the final moisture content, which is also, of course, as you can imagine, a challenge. And this is what we've been studying for multiple years. And so we looked at different, of course, metrics on those germinal vesicles after they were dried, after different times, then after different storage conditions. But interestingly enough, using just a simple tunnel assay, we noticed that actually the DNA integrity was pretty good overall, regardless of the time of the time point of dehydration, or even after storage for multiple weeks. So that was kind of a first encouraging result. Of course, we didn't stop there. We looked at other metrics like the integrity or the shape of the nuclear envelope, also the integrity and the configuration of the chromatin of the germinal vesicle during different drying times. And what was interesting is that we always, of course, compared with our standard methods of vitrification at low temperature, of course, ultra low temperature. And what we noticed actually is that we can definitely recover a much better integrity in terms of the chromatin and the nuclear envelope by drying the germinal vesicle instead of freezing or vitrifying them by cryopreservation. And interestingly enough, also the epigenetic marks that we looked at are not really different between the modifications are not different between desiccation and cryopreservation. We did that, actually, we had the opportunity to look at that in actually in different models, not only in the domestic cat. Early on, we had also some assays where we tried, to dry those germinal vesicles and to see if they were just simply viable after we would reconstruct an oocyte with a fresh cytoplast, a rehydrated germinal vesicle, trying to see if they were still viable. And we were very happy to see that, a fair amount of those reconstructed oocytes with dried, rehydrated germinal vesicle were able to resume the meiosis and to reach metaphase 2, then after that to be fertilized and to form some embryos. Of course, the percentages are very, very low, but this is again very encouraging. So more recently, what we've done for oocytes is that, well, as you can imagine, when I'm talking about the germinal, so we looked really comprehensively at the integrity of germinal vesicle during dehydration, but this is still very heavy then after that to use that, germinal vesicle and to inject the germinal vesicle to reconstruct an oocyte. So we went back to our original idea, which was kind of drying the whole oocyte, the cumulus oocyte complex. And this is what we've been doing recently. My colleague Pei-Chi Lee published this in the Journal of Assisted Reproduction and Genetics not that long ago. But in that case, instead of poking holes in the membranes of the oocytes to incorporate the trehalose, we used nanoparticles, which is a slower process, but it's also helping us to keep the integrity of the membrane. And here, those are very, again, preliminary results. But what was really interesting is that in the presence of trehalose, after using those nanoparticles loaded with trehalose that were incorporated naturally by endocytosis by the cumulus oocyte complex, we can definitely protect them from dehydration. Of course, as you can see here, the time points are not really, we are not going as far as we can go with the germinal vesicle. 20 minutes is not removing enough water to be consistent with storage at ambient temperature, but this is already a first interesting step. And also we looked at multiple types of gene expressions also in those reanimated cumulus oocytes complex after reanimated, and then looking at really the difference between fresh and rehydrated oocytes. Of course, we did some studies also in the sperm. So in the sperm, well, the sperm is kind of the ideal cell for this kind of a crazy preservation approaches, because the DNA is highly compacted, it's small, it doesn't really have a lot of cytoplasm. And then when we need to use it, what really matters the most is really the DNA plus also the centrosome. So we looked at the same approach. We did the exposure to trehalose dehydration with microwave pulses, and we were able also to, unfortunately, of course, you lose the sperm motility. So that's not something that that's something that we will have to address later on. But in terms of the morphology, there are no changes. The DNA integrity is still pretty good. And when you inject those oocytes into those sperm into oocytes, the fertilization and the embryo formation is not that bad. And we even had a collaboration with colleagues in Germany, in Berlin, where we dried the sperm in the United States. We ship the sperm to our colleagues at non-cryogenic temperature, and they were able to rehydrate them, to inject them into oocytes, and then after that to obtain embryos themselves. So that's, again, something very experimental, but very exciting. Well, we didn't stop to the sperm or the egg. We are also really interested in ovarian tissues and testicular tissue. So for the ovarian tissue, it's much more complex because, of course, it's larger. It's a tissue biopsy and different types of cells that have different volumes. So in order really to incorporate the trehalose, to remove enough water, this is very challenging and very complex, but we've made some interesting progress. So far, we are able to remove water for 10 minutes, which is showing us also that about 50% of the follicles, the primordial follicles in the ovarian cortex, can survive. And then also they have their DNA integrity is also acceptable. But importantly enough, also the cells in the tissue biopsies are able to resume also transcriptional activity, which is extremely important also because this is really the basis of everything in terms of resuming life. We have a lot of progress to make, but this is, again, something very interesting. We did the same approach for testicular tissue, and in that case, we compared actually prepubertal and adult testicular tissues using, in that case, again, the domestic cat as a model. And we obtained also very preliminary but very encouraging results in terms of the morphology of the seminiferous tubule and the viability of the cells, and importantly enough, the DNA integrity into the cells. So what we've done also more recently with my colleague Olga Melkina, and also in collaboration with Chili, we looked at a lot of different gene expression modifications before, after rehydration. And we noticed that there are a lot of differences actually between the stress, because you measure basically when you look at the gene expression, how those techniques of either vitrification or cryopreservation or dehydration are affecting the cells. You see, of course, a lot of differential gene expressions that are, of course, a response to the high stress that the cell or the tissues are going through. And we're still analyzing the data, but we published also this kind of a very comprehensive database showing all the different cluster of genes that are affected or not, either by desiccation, rehydration, or cryopreservation and warming. So that's, again, something that will help us also to probably improve our techniques and our approach, in the future. So now when we think of the future, well, we need to start thinking, of course, we would like to get out, of this complexity, as I explained at the beginning, with the liquid nitrogen tank, the alarm systems, and the possible failure. We would like to do something much more flexible and comfortable, like, well, it's maybe pushing a little bit too far, but like a pantry in your kitchen. But what you need also to think is that, okay, so easy process, storage, transport, of course, easy transport if you can, have dried samples that can be stored at ambient temperature. And then, of course, you can think of doing also the drying in a very efficient way, in the field conditions, or in countries that do not have, access to liquid nitrogen. And everything should be also in the future done at a lower cost, not only for the process of the samples, but for the storage. However, what you have also to keep in mind is that if we switch, at some point, part of our storage of biomaterials to dried collections at ambient temperature, well, it's going to be requiring new operations. We're going to have to make new plans for the management, the curation of the dried collections, and importantly enough, there will be new biosafety rules and recommendations, because, of course, those samples, they are going to be so easy to store and so easy to transport that we're going to have to be extremely careful to make sure that they are safely stored also. But in the end, this is definitely not the end of the ice age, because we still rely heavily on cryopreservation right now to preserve fertility, or for other assisted reproductive techniques. But I think it's interesting to think of these different approaches involving drying and storing everything at ambient temperature. I would like to thank my colleagues, again, Dr. Chili, who is really the person leading all the efforts in the laboratory, and Olga Melkina also, who's been working for a while with us on these topics. This is my email address. You can contact me if you have questions. We also have a profile on ResearchGates. I would like to thank, of course, the funding sources for all that research, and the main collaborators. So thank you so much for your attention, and I will be happy to take questions now and later during the panel.
Thank you so much. Thanks so much, Dr. Comizzoli. That was, whenever you see a paradigm shift of a lecture, instead of freezing, drying, that's, I think everyone is quite impressed. We're running a bit behind, so we'll just do one quick question, and then we'll circle back at the Q&A. And I think a very common one that everybody wants to know is, in your opinion, how far away are we from using this clinically, specifically in our field of IVF? What will be first, and how far away? What's your prediction?
Well, my prediction is that, well, as I said, we mainly work right now on domestic cat as an experimental model. We tried also a little bit of non-human primate materials. But I think what really conditions really the time for translation to human fertility, I think it's for me to be able to access also some leftover, biomaterials that you guys are not using for IVF, and we really want to be able to try some of those techniques for human, and to see really what is the difference with human sperm or eggs. So that's really, to me, what really will condition the speed for translation to human fertility.
Thank you very much. Our third speaker today is Dr. Amir Arav. He received his bachelor degree in animal science from Hebrew University of Jerusalem and his doctor of veterinary medicine degree from University of Bologna. His PhD degree in neuroscience, physiology, and biomedical engineering was a combined project of the University of Bologna and the University of California at Berkeley. His postdoctorate was done at the Center for Biostabilization at University of California at Davis. Dr. Arav is a leading scientist in the field of cryobiology and reproduction with over 150 related publications and more than 300 patents currently. Dr. Arav also is a voluntary assistant professor at the University of Miami School of Medicine in Florida. It is an honor to have him speak on our third lecture, The Techniques of Cryopreservation for Ovarian Tissue and Whole Ovary, Vitrification versus Slow Freezing.
Good. So I'm going to speak about cryopreservation of ovarian tissue.
Sorry, we can see your presentation, but your video is not, we can't see you.
Start my video. For some reason, it doesn't work.
That's okay. We can move forward.
Okay. So, so I'm going to speak about cryopreservation of ovarian tissue and the whole ovary. And I'll compare vitrification to freezing, especially directional freezing, but not only. Okay. So disclosure, I'm both founder and shareholder of Fertile Safe and AI Technology. And the three options to cryopreserve ovaries, slices or whole ovary, are the slow rate freezing, where we do not control very much the morphology of the ice, but we do control the cooling rate by changing temperature in a time unit. Directional freezing, however, we control a cooling rate by changing the velocity and temperature gradient. So basically, we have a temperature gradient here in the path, and we are moving through the temperature gradient in a constant velocity, so we can control the size of and morphology of the ice crystals. Vitrification, we do not have ice crystals, so it's completely glass formation of the water. So basically, it's probably the optimal way to cryopreserve tissue and organs and whole organs. So for probability of vitrification, what is important, what is the most important, are the cooling rate and the warming rate, and the probability of vitrification increase with the increasing cooling rate and warming rate, and with the viscosity. Viscosity is done by the cryoprotection concentration, so we can increase the concentration to a certain level, which is not toxic. Volume, which we just heard in the first talk, very important for oocytes and embryos, but for tissue and organs, of course, it's very limited. So I'll show you a short video of how directional freezing is. So, okay. So we started directional freezing with a whole ovary of sheep, And this was the first 4 animals that after 2 years after transplantation shows a normal endocrine function, and also we were able to aspirate oocytes and produce embryos from these animals. The record was 6 years after transplantation, and as you can see here, this is the ovary of a sheep. 6 years after transplantation of whole ovary directional freezing. You can see that there is many follicles here. This is where the anastomosis of artery and vein was. And in histology, they shows many normal follicles with oocytes inside, GB oocytes, and mature oocytes as well, and corpus luteum. So very, very nice morphology of the ovaries. But I think that a nice comparative study between directional freezing and conventional slow freezing was done in the group of Fulvio Gandolfi in Milan. And he compared the directional freezing of whole ovary to fresh control by taking slices from after warming and culture them for a week in vitro culture to show how many primordial follicles develop into primary stage. And it was interesting to see that there was no difference between whole ovary freezing to fresh control. Surprisingly, the slices, directional freezing of slices was lower statistically than the whole ovary freezing, but both were much better than the conventional whole ovary freezing, slow freezing, and also better than the slices, cortical slices, conventional freezing. So these are the results with whole ovary and ovarian slices. Now, vitrification, as we just heard, is probably the method of choice for oocytes and embryos. As you can see here, the comparative study between take-home babies from the number of sowed oocytes was like double than the slow freezing at any age of the patient. So we developed an automatic vitrification device called SARA, where we can vitrify the oocytes in straws, in a special straw called EVIT, and also in a large straws that can keep slices, or ovarian slices, of the dimension of 1 to 3 to 5 millimeter each. 6 of these straws. So we did human ovarian slices using this straw here, the EVIT, which have a mesh in the tip, so we can load cryoprotectant into the straw and automatically, and we compare after vitrification to fresh, and we at least histologically there was no differences between vitrification and fresh control. Now, as we know, cooling rate and warming rate is the most important for vitrification, and we try to increase the cooling rate by using this device. We called it VitMaster, and the way it's worked, it's produced super cool liquid nitrogen, so we reduce the temperature of liquid nitrogen to almost a solid state of minus 210 degrees centigrade, and this way the cooling rate is about 5 times, at least 5 times faster than liquid nitrogen. So here you see this is in slash, what we call slash liquid nitrogen, and this is in the liquid nitrogen itself. So temperature measurements with different size of tissue. We compare the large size of 2 to 6 to 10 millimeter with liquid nitrogen cooling to slash liquid nitrogen There was a double cooling, but still not enough, not rapid enough to allow vitrification. So the small size of tissue, 1 to 3 to 5 millimeter, produce with liquid nitrogen 4,500 degrees per minute, and with slush over 10,000, which could be okay for successful vitrification. Another thing which is important is the warming rate, and we used a new device called the RF oven system. It's not a microwave; it's a radio frequency with different frequencies of radio waves, and comparing hot air to radio frequency, we had much better warming rate of tissue and organs. Amorim, in a very nice review paper, showed that the difference between vitrification, compare vitrification to slow freezing. And there are 2 points here. One is that with vitrification, you still need to work with a very high concentration of cryoprotectant compared to very low that you need for slow freezing. But the most important is the size of the tissue, that for vitrification, you need really to go to a very small size in order to get this cooling rate, instead of larger size that you can use for slow freezing. And this is probably the reason why most of the, or all of the babies that was born after freezing after cryopreservation of ovarian tissue was after slow freezing and not vitrification. So to understand what should be the problem with large volume, we use a very large model, which is the whole limb of a rat. So here it is in a bag, the posterior leg of a rat. And we compare direction of freezing to vitrification using this. And after warming, we do anastomosis. We transplant the limb back to the animal. So you can see here the anastomosis of artery and vein. And we compared directional freezing to vitrification. This was already published in 2017. In the beginning, after 2 days or 3 days after transplantation, the tissue looks very good. Also, the limb was okay, but we could not get survival of the limb after 3 days. So the 3 days was maximum for both directional freezing and vitrification. And we decided to go and to see if we can speed up the cooling rate with the VitMaster, with a liquid nitrogen slush, and to see if what's happening. And we were lucky enough to measure temperature of different time in liquid nitrogen or liquid nitrogen slush. So if you cool for 3 seconds or 7 seconds inside the slush, you reach down to minus 150 degrees centigrade, and the tissue look okay. Also after 10 days, so POD is 10 days after transplantation, the tissue looks okay, and the histology looks very good. Both the skin follicles, hair follicles, and fat and muscle, everything looks okay. However, and this was very surprising, that when you go to 10 seconds in the liquid nitrogen slush, you reach already minus 196 degrees centigrade, and there is a massive bleeding of the tissue. And you can see here that there is a full necrosis, and there was damage all over, and the derma, epiderma, and the muscles, everything was gone. If we stay 20 seconds, we reach minus 206 and there was completely damaged. So probably if we are cooling to this area and keep and store at this temperature, we thought that we can get better survival of the tissue or the organs. And this is what we did, and this is our protocol. So very rapid perfusion through the blood vessels, and then rapid freezing, rapid cooling to liquid nitrogen slash, only for 4 seconds, no more. So you reach about minus 80 degrees centigrade, and then gradual cooling to minus 120, where you do the storage. And then after storage, warming at water bath, 38 degrees, again do a perfusion for 10 minutes, and then transplant the limb back to the animal. So this is the limb, how it's look like at the vitrified state. So it's completely vitrified, as you can see here. And this is 10 days after the limb looks okay. And this is 32 days after. So you can see hair growth and also bleeding from the fingers, as you can see here also. So there is a good circulation, and the histology looks very good. So the histology, here you can see the skin and the muscle and the fat. Everything is integrated, especially the muscle, because the muscle is probably the most sensitive tissue in this case. So this is the first animal, 35 days after transplantation. This is the limb, is the posterior limb, the left one. After transplantation, cryopreservation and transplantation, and it's look very good, and also have a sensation since the growth of the nerve, 1 millimeter per day. And you can see here the animals look very promising. So we decided to take this protocol and use it for whole ovary. And this is what we did. We use a rat ovary. In this case, as you can see here, the whole ovary, vitrified in the slash for 4 seconds, and then then store at minus 120. After warming, we just transplant it under the skin for 2 weeks, and after 2 weeks, we open and look at the ovary, cut and do histology, and you can see here follicles. And we count the number of primordial follicles and primary follicles in 1 millimeter square, and compare it. So we compared liquid nitrogen storage and liquid nitrogen cooling and storage to liquid nitrogen slash and minus 120 degrees storage. After warming, there was no any difference between the number of follicles per millimeter square, but 2 weeks later, after transplantation, there was a big difference of double the number of follicles that were cooled rapidly and stored at a temperature of minus 120, not in liquid nitrogen. So in summary, directional freezing of whole ovary and ovarian tissue is superior to slow freezing. Small size of ovarian tissue using EVIT, the straw, and automatic vitrification is possible. Large tissue or whole ovary vitrification using liquid nitrogen is damaging. And this is why we use this protocol, new protocol of vitrification, where we do rapid cooling in liquid nitrogen slash for maximum 4 seconds, and then cooled gradually to minus 120 degrees and store at this temperature. So I would like to thank my collaborator, Professor Pasquale Patrizio from University of Miami, Yal Gur from Sorotsky Medical Center in Tel Aviv, Lino Loi from Teramo, Italy, and Sergio Leda from Sassari University, and my group of fertile safe. Thank you very much.
Thank you, Dr. Arav. That was amazing. The research that you're doing is quite impressive, and I think our minds are just exploding with thoughts on how this can be applicable in so many different ways, even outside of fertility, of course. I think a couple questions for you. We'll start with one right now, and then we'll go into Q&A and open up for all of the panelists. But I hear that you're an advocate for automation. Do you think all of this, now that you've got a very good protocol for the vitrification in this manner, could this be automated?
If the automation will be the method for vitrification, I think it's very important to do automation because of the standardization of the whole process. There are so many cryo tools and so many protocols and so many solutions that everybody uses. I think that it should be standardized. This is why automatic probably will be the way to go in the future.
Thank you. Thank you. That's a great answer. I think we're good to probably open up the Q&A for everyone. So I'll let Dan find a question for us. It could be for any one of our panelists, and we'll kind of go back and forth and try to answer. There's a lot of questions, so if we can keep the answers nice and short, that would be fantastic.
Yeah, there's lots of questions, so we'll try to do rapid fire for all the presenters. Dr. Liebermann, we'll start with you. There's a lot of questions related to the culture media you use or prefer, or ones that are maybe commercially available, so someone could try to replicate this?
Yeah, I tried as much as I can, but I'm always behind answering all these questions in the Q&A. We use in-house Irvine media, but as I showed, I don't want to focus on a special brand or a vendor. It works whatever you have at home. Is it Kitasato? Is it Sage? The concentration is the same. The DS between Kitasato, Irvine, or Vitrolife, is no difference, right? There's maybe a minor difference in the molarity of the sucrose, but it's so minor. And what else, Dan?
That was good. We'll pass it along. We're going to do rapid fire. There's a few more coming to you.
I have another
Yeah,
question.
you are welcome anytime.
Lots of questions. Dr. Liebermann, Elena from the UK says, I see you use high protein post-warming. If we can't use additional protein, but only the commercially ready media, would we expect a difference in the outcome?
I answered her the question already. As I showed, we cultured also, because I'm aware of Europe has these limitations by not having 20% media available, and they cannot add as we do here in the United States or in Canada. So I tried, to culture in 10% overnight, and there is no difference.
Correct. Thank you. That's a great answer and good to know. Dan, do you have a question for one of our panelists?
Let's pass on. Dr. Comizzoli, one question we have is, since trehalose is a very expensive chemical, is there a reason for using that over a cheaper alternative like sucrose?
Not really, unfortunately. Not really, unfortunately, because trehalose has a structure also that is a little bit from conformation that's different from sucrose, and it's not possible to replicate with sucrose what we've been doing with trehalose, unfortunately.
Dr. Comizzoli, another question for you then. We have Danielle from Belgium. Daniel from Belgium. How do you select the sperm after rehydration for ICSI, especially if they've lost their motility?
Well, I mean, they don't move. Well, it's a little bit like random, as long as they look good. But there is no real. We don't really have the fancy techniques that you guys have in humans, really, to select the sperm for ICSI, especially if they're not moving.
Perfect. Thank you. That must be difficult.
All right, Dr. Arav, a question specific to you. In the case of freezing ovarian tissue that is done today, what is your professional opinion of slow freezing versus vitrification?
In case of
ovarian tissue, like current standards of ovarian tissue freezing.
Yeah, the current looks the only way right now for preserving large tissue of ovarian slices. So so I think that if vitrification with liquid nitrogen, I don't think that it's going to work with ovarian slices so far. Okay.
Thank you. This one's to Pierre again. Do we know what the explanation is for the sperm motility loss by your procedures?
Yes. Well, you know, it's pretty drastic. I think we right now we can preserve, you know, DNA or DNA configuration, but the loss of motility is explained because the fact that, you know, we probably damage highly the mitochondria and the flagellum, so there is no way we can recover that right now after rehydration.
Interesting. I'm just thinking as well, I don't know if you've ever tried this. I've heard koala sperm doesn't freeze very well, or maybe vitrify very well. I wonder if this has been done for that to see if it's more successful.
Well, we currently have a project that we are trying to fundraise with our Australian colleagues, because marsupials in general, the sperm is very difficult to freeze. So we are thinking maybe drying would be the way. Good point.
Dr. Liebermann, you've got congratulations from Stephen Fleming of doing such a leap forward in vitrification protocols. The question is, what do your findings tell us about the long-running debate regarding dilution and washing at room temperature versus 37 degrees Celsius?
Yes, of course, I looked into this too. I know there is a movement a long time ago, supported by Vitrolife, you know, to do vitrification at 37 degrees. I did that too. It works, but I think you have to be faster. Your solutions also get quite fast, more thick when you do it at 37 degrees. So I did this. I had survival. I did it even shorter, like 30 seconds ES, 30 seconds VS at 37. But is this then a very quick protocol that is really hard to handle? Regardless of your skill, of course, we expect good proficiency and good skill. But, you know, we get to a point where I think there is no gain. So I prefer room temperature for the freezing, and obviously, of course, for thawing, nothing else than 37 degrees.
Thank you, Doctor. Go ahead, Dan.
One for Dr. Arav. What, in your opinion, what is the optimum size for ovarian tissue slices freezing?
For slow freezing, as we use for directional freezing. If you use directional freezing, you can use any size, and we found out that even the whole ovary is pretty good, as I showed. For slow freezing, I think that what Morim shows of a little bit 10 millimeters on 5 on 2, I think. You can look back at my presentation at the paper of Amorim. I think that this is a good size. For vitrification, according to what we found, the only one that works is 1 millimeter on 1 on 5 millimeters. So very small size of tissue.
Thank you. I think we've got time for maybe 1 or 2 more questions. Dr. Liebermann, another question for you. There's some questions around temperature. One from Gladys says, if thawing into DS instead of TS, does the DS need to be warmed to 37 degrees?
Yeah, you know, this is definitely a requirement. I'm not challenging. You want to have a high temperature as much as possible, and that is 37 degrees. You don't want to go higher, but you should go in 37 degrees, you know, because of the warming rate, and that will be the standard. I'm not challenging this.
Perfect. Thank you for clarifying. And maybe one other question for you before we maybe jump to one last one. What is the equilibration time that you recommend for embryos and oocytes, post thawing? Is there a preferred time?
So equilibration for vitrification.
Yeah, so post warming.
Post warming, you mean going to ICSI?
Yeah, before utilizing either the oocytes or transferring your blastocysts.
Yeah, we usually give them 2 to 3 hours for oocytes, right? But if you ask about transfer of blastocysts, I would stay in a 2 hour, 3 hour window. We had cases, it depends on our schedule. Sometimes, we have 15 FETs and we thaw early, and when we have blastocysts, as I showed, 4 hours later, they still do fine. If you don't have a big volume and you can schedule them earlier, I would transfer them in 1 or 2 hours.
Perfect. That's helpful. All right, Dan, do you have one more question?
Yeah, let's do one last one. So Dr. Comizzoli, this comes from Philip Chan. First of all, excellent presentation, of course. Are the pulses and perhaps frequency wavelengths of microwaves used for assisted dehydration different for different cell types like sperm, eggs, or embryos?
No. This is just the same. The difference is just the exposure time to the wavelength, and there is no need to change from the frequency are exactly the same for every single biomaterials that we used. Something that I wanted to add also is that when we talk about preservation at room temperature, that means that also there is a flexibility, there is a range of temperature that the samples can be stored with. I mean, it can be 25 degrees Celsius or 12 degrees Celsius. Then that's the beauty also of the technique, is to have kind of a flexibility into the temperature and not being constrained to a fixed really low temperature. Thank you.
Amazing. Thank you. We look forward to learning more on this as your research continues. I think we'll wrap up. That is time. We've heard some wonderful talks. I want to mention that this webinar has been sponsored by Future Fertility. We would like to thank everyone who took part in this webinar and found time in their day today, as well as thanking my co-hosts, Dr. Dan Nayot, Dr. Juergen Liebermann, and Dr. Pierre Comizzoli, and Dr. Amir Arav for your talks today. Thank you.
And of course, we can't forget our team in I3. Thanks for the organizing committee of this whole initiative of I3, Jacques Cohen, Peter Nagy, Marianne Savete, Thomas Elliott, France Farley, Gilles Palmer, and Violetta Sura. Our next session will be March 12th. It's the return of RBMO Live. This is a really exciting edition with choice papers and some key tips on how to get your paper in. But caution, there's a little bit of a time clock change. So it's going to be 3 p.m. Eastern time. That's not always the same for the UK. It might be 7 p.m. or 8 p.m. So just clarify, but we will see you March 12th for our very next session. Thank you, everybody, for the I3 team.